So, at the point of formation 2, if we have that 180 liters, basically that proximal convoluted tubule reabsorbs about 2/3 of what is in the filtrate. So it's a pretty busy area of the nephron. Osmosis is a major player in the PCT. And then something that we're going to find in the cellular structure, we're going to find something called an aquaporin. So that aquaporin we'll find in these cells, and guess what's the only thing they deal with? Water. So they can actually increase and decrease in their number if they need to. The cells will do that, which is kind of cool. So urine formation 2, we get to the point where the biggest goal, help return the water, help get electrolytes back. So this is what would happen with the cellular structure and the relationship to the blood vessel. The materials that are going to move between the cells, they're going to function based off of that solvent drag. So if we have the materials that are going to follow water, for example, we're going to see the urea, uric acid. Look at everything that can follow this, okay? We can have sodium follow, but the big players are going to be the potassium, phosphate, and some of these other salts that are needed for the functions in the body. If we look at what's going to have to go by way of making it across the cell, this is where the function of those proteins become important, because one of the substances that does not follow solvent drag is glucose. Okay? Glucose does not follow by solvent drag. We can't lose the glucoses. Why is it so important to get the glucoses back to the body? And what's the biggest, well, I wouldn't say the biggest organ, but the brain. Okay? So we really can't lose the glucoses because of the brain function. So, in looking at these transport proteins, this is that little aquaporin. Simply water just goes, and that's it. Nothing else, okay? But these other transport proteins, for example, the sodium. If you remember, I said glucose wants to follow sodium. So as a sodium moves across the cell membrane, a glucose will move with it. Now watch, this is our transcellular route. So that means it came in on the apical surface, but it's got to leave by the basal. So that glucose has to make its way all the way across and exit out of that basal surface. That's the only way that we can get those glucoses to move. So if this is not functioning correctly, the glucoses are going to continue to move. They're going to go, because once we get past the proximal convoluted tubule, what's the next portion that we're going to hit? We are going to hit our, and I don't really want to lose this, okay? So now we're going to hit that descending limb of the nephron. Now, if you remember, the nephron does this number, okay? Some of the tube is big, some of it's skinny, some of the tube is big, okay? And we're going to have a certain cellular structure that's going to be present. And when we look at the two types of nephrons, we had some of these little nephrons that if we were looking, if this is my cortex and this is my medulla, some of these little nephrons only dipped down a short amount. And then some of those nephrons dipped down a little bit further. The ones that dipped down a little bit further, they had the capillaries around them. Do you remember that? And they only made up 15%. The ones that dipped down, no capillaries, were the 85%. Okay? And then around the glomerular capsule and all of their convoluted tubules, they had that huge ball of capillaries. Do you remember that? All right. So, at this point, okay, we've got these two different structures of the nephrons. In the nephrons, we're going to see a little bit of a different structure on this descending and the actual loop, and then the ascending as it goes up. All right, because we've got the tube that's going to be a little bit thicker, gets thin, and then gets thicker again. Each part, if we're looking at the one that's dipping way down into the medulla, each part is going to have the ability to allow for something to move, some type of material. So we move into, did I miss a mark? Hold on, I think I got a typo. Okay, no, but I do have a typo. Because there's 4 stages, okay, but still in my urine formation too. Peritubular capillaries, okay? Remember how in our nephrons, capillaries or capillaries, okay? So, peritubular, I'm looking at those capillaries that would be close to my tubes that are present, okay? Those capillaries can reabsorb water and the solutes. So in that proximal convoluted tubule, the water that got to move across, that gets to return to the blood, and then my electrolytes get to return to the blood. Because at the point that we're at, those are the biggest movements. And then, of course, my glucoses. Okay? We know that it moved by osmosis, and then all of those little electrolytes that followed the water between the cells. Now, there are factors that affect the movement of the water. If the water is moving, that means the electrolytes are going to move. But wait a minute. Is urine always consistent? No. Sometimes it can be more dilute. Sometimes it can be more concentrated. Do what? Yeah, it depends. It depends on how hydrated you are. Okay? So, and then, there are some other factors that can affect that too. Tissue fluid pressure. The actual tissues themselves, all right, can drive the water into my blood vessels. We have blood water hydrostatic pressure based on where we find it, okay, can affect the movement of the water into the blood. And then do you guys remember me mentioning the colloids? Those actual particles that are moving, okay? The higher the particle, the lower the water, okay? Once again, water wants to go from an area of low concentration to high concentration. Yeah, thank you for correcting me. Okay, so the osmosis movement. All of these factors, they're working to move the water into the capillaries. We don't want it continuing through the rest of this nephron into the collecting duct into the bladder, because then we're going to have a high, amount of urine that's being produced, and we don't want that. The normal production, about 1 to 2 liters. Now, in times of blood pressure problems, okay, high blood pressure, low blood pressure, remember us talking about those, okay? And remember, when we talk about blood pressure is dealing with the heart and that pumping action of the heart, the strength of the contraction of the heart, and the amount that's moving through the heart. Under normal conditions, this is happening all the time, okay? However, in certain cases, it becomes chronic. Remember that renin-angiotensin that's going to be, it's going to actually have to use the respiratory system? When that system takes place, if renin gets released, makes its way to the respiratory system, gets converted into the angiotensin, and so on and so forth, that is going to affect these pressures. Under normal conditions, the pressures want to favor water going back to the blood. Okay? That's the goal. In the plasma membranes, there is a limited number of the transport proteins. Now, if needed, cells, okay, can create more, and then they can destroy them if needed, okay? But the number is limited. If my number of transport proteins is limited, does that limit the movement of the solutes? Yes, because they can only function to the ability of what they have, okay? So this is a mechanism that will affect transport of those materials. If all of the proteins that are available are being used, that means materials are going to continue to pass by and not make it across or between, well, not necessarily between, but across those cells. All right? So that tells me that the solutes could simply pass by all of these cells. And if they do, they are then going to make their way into the next segment, nephron, into the distal convoluted tubule, and so forth. This is what happens with diabetes. In diabetes, what is the major problem that is associated? Insulin's not functioning correctly, so the glucose is not being used correctly. So a lot of glucose makes it to the nephron. And because there's more than what can be handled by the transport proteins, the glucose continues the descent through the nephron and therefore makes it to the urine. How did they originally test for diabetes? The first test, they tasted the urine. And if the urine was sweet, the person had diabetes. Yep. So, that's what can happen if too many of the solutes move past these transport proteins. Now, to me, the terms are backwards, okay? But then again, if you stop and think about it, it's like, oh, okay. Tubular secretion. If you think about secretion, I think about something moving out of something, okay? Like, I don't know, something moving out of my skin or whatever. I don't know, okay? But look at this. It's just backwards to me. That renal tube, this portion here, extracts chemicals from the blood. Okay, so here now to here in this renal tube and secretes them to this tubular fluid. It does seem backwards, doesn't it?
But it's secreting it to the next layer of tubes, not to...
And once you think about it, it's like, oh, okay, I got that. So what is the purpose of the tube doing this and the nephron? Acid-base balance. We need the blood's pH to remain roughly that 7.4. All right? Waste. Even though we've got the blood entering the kidneys, it can still be waste in the blood in those capillaries, and we need to get it out. Clears the drugs and contaminants from the blood.